Welcome to AP Biology. We're starting a unit now on transcription and translation, how to go from a gene to protein. And we're going to actually spend this video on transcription. So the overall view, so here's where we are today, but the overall view is that DNA codes for RNA. We're going to be talking eventually about making a polypeptide, so we're going to be talking about messenger RNA, but DNA can also be transcribed into other types of RNA, like transfer RNA or ribosomal RNA or some others. But for this unit right now, we're going to be talking about messenger RNA. So there's a pre-RNA, and then that gets modified a little bit, which we'll talk about later in a different video, and then that RNA will be translated into a polypeptide. But for right now, we're just going to be talking about transcription from DNA to RNA. So how does this happen? Well, there's an enzyme. In biology, pretty much every step of every reaction needs an enzyme. And so RNA polymerase is really well named. ACE means it's an enzyme. RNA polymerase means it's an enzyme that makes an RNA polymer. So here's DNA, and DNA makes RNA. How does it do that? Well, it uses this enzyme. RNA polymerase. DNA polymerase, you might remember from the replication unit. It's one of the enzymes, one of many enzymes that you need to make more DNA. We're not talking about making more DNA. Now we're talking about DNA making RNA. And the enzyme used to do that is RNA polymerase. So it separates the DNA strands and makes a complementary RNA polymer. This is so whenever we're talking about these, remember there's this physical molecule, this big thing here that separates the DNA and actually reads this side of the DNA to make an RNA molecule. When we talked about replication, there are a ton of different enzymes. I think RNA polymerase deserves a ton of respect because it does tons of stuff. It doesn't need a different enzyme to unwind it or to clip it or to do whatever. It just goes ahead and reads it. Okay, so RNA is made complementary to the template strand of DNA. Same base pairing rules, except you use uracil instead of thymine. So here's your DNA molecule, and it's double-stranded, and you're reading it to make RNA. So see how RNA AUG. If I didn't show you this up here and you saw AUG, I would want you to look at these two different sides of DNA and be able to tell me that this is the one that was read. So RNA polymerase saw the T and put in a matching or complementary A. It saw the A and put in the complementary U, saw the C, put in a complementary G. The side of the DNA that RNA polymerase actually physically reads is going to be called the template strand because it's used as a model or a template. And the other strand is called the coding strand. So why is it called that? Well, if you look at this stuff, do you see how the A here, it's the same? And okay, the T's are different, but you've got G then A. Oh, I have that here too, G then A. I've got C here. Oh my gosh, this whole thing, CG, it's exactly the same, couple of A's. The only thing that's different here between the coding strand of DNA and the RNA is that whenever there's a T in the coding strand, we use a U, the uracil, instead of the thymine. So the coding strand looks exactly like the RNA transcript, except for the T's. So you need to memorize template strand compared to coding strand. Template is what the RNA polymerase, this enzyme, physically binds to and then forms these complementary pairs. So it reads this side of DNA, the template strand, in order to make the RNA. So the template strand is the same for a given gene. So let's see if I get this right. Yep, okay. So let's say the one in dark yellow that I'm using here, this is the template for this particular RNA that's made, but it won't be the template for some other gene. There'll be some other gene somewhere else that maybe uses the other side. Right? So the template strand is always the same strand for one particular given gene. It doesn't hop back and forth. Okay, you're going to love this part. So we had said that the template strand is the strand that RNA polymerase reads, right? So the other strand, they have all these names, which you're going to love. And AP actually wants you to know all of them. But look, template strand, the opposite is the non-template. Kind of makes sense, right? What I memorize is template strand and coding strand. So the coding strand looks the same, except it doesn't have T's, right? It has U's. And if this is the coding strand opposite to the template, right? If that's the coding strand, the other one's the non-coding strand, almost makes sense. The coding strand looks the same as the RNA, so they also call that the sense strand, which means the template would be the antisense. I hate that there's this many names for it. There's the plus strand, which is the coding strand, and the minus strand is the template. I hate them all. I hate that there's that many words for you to memorize. But what I will tell you is if you memorize template strand is the strand that you actually read as a template. The RNA polymerase actually looks at that, sees sees like a T and puts in an A, sees a G and puts in a C. If you memorize template strand and the other one, if you memorize coding strand, you should be okay. I think you'll be able to make sense of the rest of it. Okay, there are 3 stages of transcription: initiation, elongation, and termination. So initiation is the start. And so imagine being initiated into like a club or something, right? Elongation is how the RNA gets longer. And then termination is a stop. So for transcription, there is the start, the middle, and the end. And that's really this: initiation, elongation, and termination. We're going to actually see these same vocab words for translation, but we'll cover that later. So right now, transcription. So here's the DNA. Super important is this, this enzyme, RNA polymerase, that reads it in order to make the RNA. So there's the initiation, the elongation as it gets longer, and then finally stopping. So let's go through those. So initiation, meaning the start of transcription, the enzyme RNA polymerase binds just before the gene. So here's a piece of DNA or a gene, right? The gene can also be called the transcription unit. It's the part that's going to be made into messenger RNA or be read to make messenger RNA, right? So here's the RNA polymerase, that's the enzyme, ACE, that's going to do the job of making the RNA. So how does it do that? Well, it has to start at the beginning, right? So here's the start point where it actually reads the DNA to make the RNA. But just before that, well, part of it also, the DNA, part of the DNA is called the promoter. So the promoter is a segment of DNA that comes just before the part that actually gets transcribed, including a little bit that does get transcribed. And it's the part of the DNA that RNA polymerase can bind to. So promoter is crazy important. You absolutely have to memorize that one. So RNA polymerase first has to bind to the promoter, which is a section of DNA that includes the transcriptional start point right here, and also several dozen nucleotide pairs upstream from the start point. So think of a river, the water's going downhill, right? Upstream is back there where you started, right? And so here's where you start, upstream is before that. So here's RNA polymerase binding to the promoter. If for some reason the RNA polymerase cannot bind to the promoter, then you're not going to be able to do any transcription. So that part's really important. Promoter is something to memorize. Some of this is fun facts, but promoter you need to know. Okay, another thing you need to know is these things called transcription factors. These are proteins that are needed to help RNA polymerase bind to the start, to the promoter, and to start transcription. So here is the gene, the piece of DNA that's going to code for the RNA that's going to get made. And so here's the very start point right here. I guess I should have brought it up here. The promoter is just, I was going to say just before the gene, but it actually includes a little bit of the gene. So here's the promoter. Promoter, again, is a piece of DNA where the RNA polymerase binds. These are transcription factors. They are proteins that help the RNA polymerase to have a nice affinity, to have a nice ability to bind to the promoter. If the transcription factors are not there, then RNA polymerase will have a tough time binding, and then you won't make very much RNA from this particular gene. So to get started, you need these transcription factors. There are general transcription factors, and then we'll talk later about specific transcription factors. But every gene needs these transcription factors to bind. The way you regulate this is if you can stop those from binding, then you can stop transcription, which means you won't make the final product. So transcription initiation complex. Transcription is DNA to RNA. Initiation means to start, and complex means a bunch of stuff. It's the transcription factors, these things, these proteins. And then it's also RNA polymerase, this thing, the enzyme that does the job, which is also a protein, right? And it has to be bound or bonded here to the promoter, right? You might notice this part of the promoter called the TATA box. T and A they have 2 hydrogen bonds. Those complementary base pairs have 2 hydrogen bonds, whereas C and G have 3 hydrogen bonds. So it's probably, I'm guessing the reason that there's a TATA box here is that it's easier to separate out those A's and T's. So this is conserved in eukaryotes. It's a DNA sequence in the promoter of eukaryotes that's found before the transcription start site of a gene. It's part of where RNA polymerase binds. So the TATA box defines the direction of transcription and indicates the DNA strand to be read, as in which DNA strand is going to be read, right? Which one is the template versus the non-template. And the transcription factors are able to bind to that. Okay, elongation of the strand means you just continue. RNA polymerase walks right along and keeps adding on to the 3' end. So if this is the 3' end, you're going to add another one there and there and there. So you can keep adding on to that. So RNA polymerase moves along the DNA, it untwists the double helix, it adds RNA nucleotides to the 3' end of the growing RNA molecule, just the same as in DNA replication. So a transcription unit is the stretch of DNA that's going to be transcribed. So that DNA is the piece that's going to be coded for RNA, so it's the gene. The gene can be transcribed simultaneously by several RNA polymerases. So maybe we're in the pancreas and insulin needs to be made, a lot of it maybe. Maybe there's a bunch of RNA polymerases reading this gene, so you're making a ton of RNAs. So you're doing a lot of transcription because you really need to express this gene a lot, maybe. Okay, termination is just the end. So in bacteria, the RNA polymerase stops transcription at a particular sequence called the terminator sequence. In eukaryotes, there's a different way. It actually goes way past, but then another enzyme cuts it and sort of eats it and eats up the extra RNA until you get to a place where it falls off. Okay, the primary transcript is the pre-RNA. It's the initial RNA transcript made. So here we are. We've done transcription. We have this messenger RNA made. It's not quite messenger RNA because we've got to do a few things to it, and that is what we'll look at in our next video.